All right, welcome back. My name is Mary Gehring. I'm going to be chairing this afternoon session, which is filled with longtime colleagues of Gerry's. We have a surprise addition to the program, so we're fortunate today to be joined by the founding director of the Whitehead Institute, David Baltimore, and he is going to say a few words. Thank you, David.
Thank you. It's an enormous pleasure to be here and to congratulate Gerry on his graduation. I've never seen so many people in this room. Gerry, in writing this book, has on the front cover a radical experiment. And in a lot of ways, the Whitehead Institute is a radical experiment, or was a radical experiment in its day. But one of the ways it was building collaborative science around multiple laboratories. And that doesn't sound like much today, because if you walk into a new lab today, it's entirely open. It's just space with benches and stuff around, equipment that many people use together. But when I first came to MIT as a student in 1960, in Building 16, it was a warren of little rooms. And a given professor had a bunch of rooms that he or she, mostly he, owned. And the idea of sharing space, the idea of coming together in multiple groupings nobody thought much about it. And when I came back here as a faculty member in 1968, and set up a laboratory. And then my old friend from Rockefeller University, Harvey Lodish, came to the MIT faculty. And I suggested that we have a joint laboratory, which in fact we set up. That was something new. The idea that two professors would not have their own dedicated space, but work together. And we had joint students, and we had joint papers, and so when we built the Whitehead Institute, it was to find faculty who could interact with each other and make and bring together ideas. And as I thought about whom I knew in the scientific community, who seemed like an exciting opportunity to interact with, it was the group that became the founding members of the Whitehead Institute. And in particular, Gerald Fink, who was at that time in Cornell, but who had come here as a visiting professor, and whom I knew from his time spent at MIT. And I couldn't imagine anybody who would be more important to bring in to the then nascent Whitehead Institute. And of course that's worked out. And we've seen 50 years of development of this institution. Some of that under Gerry's leadership, all of that with Gerry's involvement. And even though I don't recognize the color of this room anymore, did you do this, Gerry? No, you don't know. I just couldn't pass up the opportunity to come back to this wonderful home for science, home for scientists, that we built on this corner, and that was sort of the nucleus from which so much else has developed around MIT. I just want to thank all of you for the contributions you all have made. I see people I've known for years and years around. And again, thank Gerry for making the leap into what was then just a nascent idea, but that has become one of the most honored institutions in American science. Great.
Thank you, David. Next, I'd like to invite up Harold Varmus.
Well, thank you very much, and hello to everybody. Nice to see so many friends out here. I'm here to talk about Gerry's earliest years because we shared them. I know Jeff has told us that Gerry insisted that there be themes. The themes here will be humor, energy, and loyalty, all three of which are going to be featured in the things I have to say. In the morning, we heard a lot about what Gerry had accomplished and what his progeny have accomplished, and here we're going to talk more in this session about what Gerry is like. Now, there are people here who know Gerry better, deeper than I do. Rose lays among them. But there are very few who've known him longer. Probably nobody's known him longer because our acquaintance goes back nearly 80 years. That happens in part because of geography, in part because of some personal dispositions to certain activities. I'm going to begin. No, we don't want this. I'd like the first slide, please. For those of you who are not familiar with the geography, this is Long Island. And something should happen here when I push. There it is. And this is the town where Gerry and I grew up, Freeport, New York. Now, we're not the only two people in this audience who come from Long Island. You just heard from David Baltimore, who came from the North Shore, about which nothing more will be said. Culturally, ethnically, in every way different from the South Shore. But today we're going to concentrate on the South Shore. Now, Freeport is a small town founded in the pre-revolutionary era. Had, as the name might suggest, an advantage to people who were trying to bring liquor in during Prohibition. But a fairly small town with a generally lower middle class population. And yet within that small town, you can see two initials here, my house on Merrick Road. Gerry's a little far north of that. We were just a few blocks apart. We were separated by the Long Island Railroad, so we were on different sides of the tracks. Nevertheless, there's a certain homogeneity in Freeport that made it difficult for me to accuse Gerry of coming from the wrong side or the right side of the tracks. Indeed, there was a lot of similarity in our family life. We both were the sons of physicians who had urban roots. practiced medicine in Freeport. They showed signs of public service. My father was the Jones Beach State Park physician. Gerry's father was affiliated with the New York, was for the Freeport Fire Department. So there were definite similarities. And then there were many things we did with our dads that were similar. Both of us grew up with a love of saltwater fishing. My father took me out to Fire Island Inlet for fluke. Gerry's father had a small boat, and Gerry was out saltwater fishing in the bay. And of course, we went to school together. But before that happened, we had another interaction, which Gerry may not remember, but I do. And that occurred roughly in 1947 in the spring when he and I were part of a host of 7- and 8-year-olds who were trying out for what might now be viewed as Little League baseball. Gerry was the star. They were all talking about that Fink kid who was running around the bases, and I was consigned to sitting on the bench in some unfortunate team. But Gerry already was distinguishing himself as a scholar athlete at a very early age, and that image was very memorable to me. We went to separate elementary schools because of the several block distance between our houses. But we then were together in Freeport High School. I was a year ahead of Gerry. We shared a pretty substantial degree of contempt for the kind of education we were served in Freeport High School, but we had smart friends, and we both enjoyed being at a school with some smart kids battling with much less smart educators. So I don't have time to review all the things that happened in high school, but I can give you a bit of a snapshot by looking at, thanks to the internet, to the way we were portrayed in our high school yearbooks. Here's mine from the 1957 issue of Voyager. Here's Gerry's from the year that follows. You'll see there are many similarities. We had different nicknames, but we both worked on the high school newspaper flashings. We both, they make reference to our athletic accomplishments. Gerry's were much more advanced than my own. And both of us are alleged to being destined for life in medicine, being the Jewish sons of Jewish physicians in Freeport. Gerry's future lies in the field of medicine. I'm going to make a dexterous surgeon. As you know, neither of these came to pass. Now, the fact that we both went to this high school allowed an event to occur roughly 50 years later or so, when Gerry and I were both informed by the officials in Freeport, New York, that we were going to be inducted to the Freeport High School Hall of Fame. Now, why does everyone think that's funny? Anyway, as Gerry and I were planning to attend this event together, the event proved, I have to say, be quite disappointing because they invited us to speak in an audience in front of an auditorium with 2,000 seats, but they neglected to invite any students. So there were roughly 2,000, or a little less than 2,000. Gerry's family and my family were there, but generally speaking, there were about 1,900 empty seats staring us in the face. At about that time, Gerry took me aside and he said in a voice that I think many of his trainees will recognize, he said, "Harold, do you know who was the most famous graduate of Freeport High School?" And I could see from his face that I thought he thought that I was going to think it was myself. But I had the feeling that Gerry knew someone that I didn't know. It would be embarrassing to me to admit I didn't know it. And indeed, the year after us, a member of the class was Lou Reed of the Velvet Underground, and I knew very little about who Lou Reed was, but Gerry, it turns out, has told me stories about his adventures at Lou Reed's house, and it's interesting that unlike me and Gerry, who had plans to become physicians, Lou had no plans. Will take life as it comes, and it did come pretty forcefully. Okay. So, as I mentioned, I was a year ahead of Gerry. We now call that a year older. But I headed off to Amherst College, remained in touch with Gerry, and I was glad to find in my roamings on the internet in preparation for this event at least 2 of Gerry's accounts of my contributions to his choice to end up going to Amherst College as well. Gerry testifies to our propinquity, and then he followed on. And I was amused to find that he quoted me as telling him that he should go to Amherst so he wouldn't just end up being another grind in Harvard Yard. And indeed, Gerry did come to Amherst. And the Amherst years were interesting, but our interests and even our stature tended to diverge at this point. Gerry was known for being an assiduous biology student and a basketball star at Amherst. I had deviated from my life as a prospective pre-med, majoring in English and writing about Charles Dickens and running the school newspaper. But even more profound was the difference in our social affiliations. So Gerry was a member of the austere, perhaps the most famous fraternity at Amherst College, Amherst Delta Phi, which was home to all the great scholar athletes. Its most memorable alumnus was Harlan Fistone, who became the Chief Justice of the Supreme Court in the 1920s. I was a member for a year or so of a somewhat atypical fraternity house full of dissident intellectuals. I'm looking for someone who was famous, who was a member of Alpha Theta Xi. I mentioned John Abele, who some of you may know is the head of Boston Scientific. He's now in his late 80s, the richest person in Vermont. And there are rich people in Vermont. In any event, these were ways in which our aura was somewhat different at Amherst College. And indeed, after college, we did go our own ways. And Gerry, as you've heard, off to Yale Graduate School and then to Cornell. I went off to graduate school in English for a while, then went to medical school. There was a long period in which Gerry and I really never interacted. Strange, perhaps not of great emotional or intellectual significance, but nevertheless interesting to me, is that we missed an opportunity to reconnect. In 1966, on June 28th, I believe it was, I was at the NIH interviewing for positions as a clinical associate at the NIH to avoid being put into the Army and sent to Vietnam. I was going from one appointment to the other in the third basement of the clinical center when I saw a person who looked just like Gerry race by me in a white coat carrying some tissue, some Petri plates. I said, God, that looks like Gerry. And it only was later when I realized that Gerry was no longer at Yale, but had moved to the NIH to work with Bruce Ames on histidine pathway. That was an opportunity missed, but it reflects a certain way the divergence in our career and the lack of contact. And over the next many years, I followed Gerry's career with interest, read many of the papers that people were describing this morning. But we suddenly had a reunion when the Bucha-Garfinkel paper on retrotransposition of TY was published. I was then working on the generation of proviral DNA from retroviral RNA, and we both were dealing with long terminal repeats and retrotransposition. And Gerry and I started to appear on the same speaking podia together, and that was interesting and gratifying. And we were in touch frequently about that. Then I came here and had a sabbatical at the Whitehead in the Weinberg-Baltimore axis. I don't have an alumnus ribbon on my, I'm a little disappointed by that, but I hope the organizers will try to rectify it before I go home. And then we retained our interest in fishing and had a number of episodes, especially one particularly memorable outing not far from his home on the Cape, but the details there have to be relegated to another session. Now, of greater significance, we found we were converging on policy matters. Maybe this was, in some respect, inherited tradition from our parents, who were socially advanced and contributory to social advancements. Gerry, as you've heard a couple of times already today, wrote the famous Fink report on how to manage publication of information about the microbes. I'm working for the Academy on dual-use research of concern, but perhaps even more influential moment from my perspective was Gerry's contributions to public understanding of science. And I want to conclude these remarks by showing you Gerry in action in 1992 when he came to UCSF, where I had organized a symposium called Winding Your Way Through DNA. The tapes of that event are preserved.
I think there's a larger problem that perhaps you have alluded to here, and that is it simply the exigencies of the marketplace in the Western world which dictate how recombinant DNA is used? And I don't think it's a simple answer, because part of it is yes. But the other part is that I think we're hiding something. I mean, it's how deeply felt is the need for vaccinating people against tuberculosis and leprosy. The answer is, in this country, when it starts to strike the people you know. And I think that, unfortunately, unless we take a bigger and wider view, we can't blame scientists, we can't blame our politicians. We live in a democracy, and we have to ourselves, as citizens, vote for people who advocate not only more money for technology, but taking a broader view of how that technology will be used.
So those are words that we can echo today. Thank you for listening to me recite the story. I hope I've brought you some elements of the humor, the energy, and especially the loyalty that Gerry has always manifested to his students, his friends, his family. And Gerry, thanks for having me here and allowing me to enjoy the celebration with you.
It's now my great pleasure to invite to the stage former Whitehead member. Excuse me, sorry. I jumped ahead in the program. No, I would like to invite to the stage Bruce Stillman, President and CEO of Cold Spring Harbor Lab.
When I got the invitation from Ruth, it was a distinct pleasure to be able to be here and celebrate Gerry's career in science and all that he's done that you've heard about in science, but also in administration. I'm going to talk about an institution on the aforementioned North Shore of Long Island, different from Freeport, and it's a place where the Fink family spent quite a lot of time. Cold Spring Harbor Laboratory was founded in 1890 as a teaching institution. In those days, mostly high school teachers and some college professors came to take courses at Cold Spring Harbor. But in 1945, Max Delbruck started the really famous phage course in a building at Cold Spring Harbor that later on became the home for the yeast course. And Jim Watson became head of the laboratory in 1969, and he was a student with Sal Luria in Indiana, and when Sal Luria and Max Delbruck used to get together in the summers at Cold Spring Harbor every summer, one time Jim Watson came, and although he wasn't part of the phage course, he hung out with those people, and then appreciated the importance of those courses. When he became director in '69, he immediately asked Gerry and Fred Sherman to teach a course in yeast genetics, which he had written about in his famous textbook. So Gerry started with Fred the yeast course in 1970, and you've seen some of these pictures before. I picked this one on the right because this big tie who I read in an article that she wrote through taking this course with Gerry that she ended up getting an offer at Cornell as a faculty member. And so taking courses at Cold Spring Harbor can pay off, in addition to learning about technology. Here's a couple of other photographs. You've seen one of these before with from Fred, but from the one on the left is shows the enthusiasm that I think yeast people had at Cold Spring Harbor. And the courses at Cold Spring Harbor, the yeast course is still going on. It's in the Delbrook building, which is where the phage course was held. And there's a bunch of things in addition to the academic side of these courses that happens. There's now a famous plate race where all of the yeast course and the bacterial genetics course accumulates enormous amounts of plates. And the courses are required to carry 40 of, the students are required to carry 40 of these plates stacked up and run around a road without dropping them. And if you have dropped them, you have to go back and start again. And there's 3 courses that have a race, and it's quite an event at Cold Spring Harbor because the whole laboratory shuts down for one day for this event. The other event was a run from Cold Spring Harbor Laboratories, Sogmore Hill and back, 9 miles. I did this a few times and it almost killed me. And here's Gerry in the middle, and Tom Broker, who, along with Joe Sandbrook, used to race each other to win this race every year. It was a grueling race. Then Gerry was a trustee of Cold Spring Harbor Laboratory for a few years and contributed to help build the laboratory up extensively during this period in the mid-1990s. And for all of that he did in teaching the yeast course and as a trustee of Cold Spring Harbor Laboratory, we were very pleased to offer him an honorary degree of Doctor of Science in 1999. And of course, he talked about education. And that was really fantastic that we were able to honor Gerry and his career for his contributions, not only to the yeast field, but to lots and lots of areas of science, including this institution here. He also attended many meetings at Cold Spring Harbor. There was annual, every other year, a yeast meeting. There's a special meeting. I think this meeting on the right was a celebration of one of Jim Watson's milestones at Cold Spring Harbor. And then there was also a meeting. We have a series of meetings on the history of molecular biology and genetics. And we just had one that Harold Varmus organized on the history of cancer genetics. But this one here was on the history of yeast and the impact of yeast genetics and yeast as a field in science in 2019. And there was a panel where here's Gerry talking about how important his favorite organism is. And that was a very good, a really fantastic meeting, and these history meetings are up on the web. Now, Gerry had influences beyond teaching yeast geneticists or people who wanted to learn to do yeast genetics, how to do yeast genetics, including how to do back crosses and tetrad analysis and things like that. I went to Cold Spring Harbor in 1979, and of course the yeast courses were happening there, but also Jim, with Gerry's help, recruited a yeast group there of Hickstra, Thurn, and Clark, and they were working on mating type regulation. And I started working on DNA replication using viruses, SV40 virus and adenovirus. And we were working on, in the mid-1980s, we were working on the SV40 system and had purified quite a lot of human proteins that were involved in the replication fork, but they used SV40 T antigen. So John Diffley, who was in my lab at the time, who was working on adenovirus, he and I had a number of conversations, and we said, well, what is the T antigen equivalent in human cells or in eukaryotic cells? And so we began to think about how to approach this problem. And I was aware, partly through the yeast course, because I never took the course, but I used to go and listen to the lectures. I was aware of the existence of these autonomously replicating plasmids and a genetic sequence that had been hypothesized to be the origin of replication, but at that time not yet shown. And so we thought, well, maybe we could work on yeast. So I went down to the yeast course on a couple of years when we were thinking about this and talked to lots of the visitors. And every single one of them told me not to work on yeast because it was full of proteases and you couldn't do biochemistry. Well, one of the things that a good scientist do is sometimes don't listen to advice. And so we worked on yeast. This is an unpublished figure from John Diffley's laboratory of all of the proteins that now can completely reconstitute in chromosome replication in vitro and chromosome cohesion in vitro. These proteins are all purified from yeast, a few of them from E. coli. So that's a lesson for that I talk to students is sometimes you have to go with your own ideas about how to approach a scientific problem. Now, what's remarkable about this is that we now know the cryo-EM structures of every one of these proteins on this slide. And so John and I and a videographer who's a cell biologist at the University of Utah have been getting together to work on making movies of this. And the first movie that we've made is on the initiation of DNA replication, the formation of what's called the pre-replicative complexes. I should add that Big Tai, who took the yeast course, went on to make many mutants in now what are called the MCM proteins, which you'll see at the end of this are the whole purpose of ORC and CDC6 loading the MCMs to form pre-replicative complexes. And so this is actually based on cryo-EM structures and intermediates that we've been able to do time courses. These structures have all come from my laboratory with Hui-Ling Li and Li Mo Joshua Tor, and with the Zai and Tai laboratories, and also the Costa and Diffley laboratory. And so this is now down to literally the atomic level of understanding this process. And that's the famous MCM double hexamer that is the formed and then is the start of replication of the entire genome. Now, another story related to Gerry is that I started to become interested in the evolution of DNA replication. When one works on a field for a number of years, one gets interested in evolution of the processes and in particular evolution of origin specification. And you'll notice in the gray that there's a bunch of yeasts represented by Saccharomyces that has defined origins, and we know the atomic reason why those defined origins, and we highly sequence-specific origins, exist. And what was interesting to me, because I spent so much time with the mating type group and listening to the people in the yeast course, was that this defined origins co-evolved with the appearance of SERB proteins that are silent information regulatory proteins, because those species had lost RNAi. And then when reading about this, I also came across a paper from Gerry with Dave Bartel where these species in between the RNAi species, most eukaryotes have RNAi, but there are a few that, some of the budding yeast at the top don't. They have the SERB proteins, but there's a bunch of them in the middle which have kind of in the process of losing RNAi, and Dave Bartel and Gerry worked on Candida albicans to show that actually it does have RNAi, but it's not used the same way. But there was one yeast that was of interest to me that didn't have RNAi and apparently didn't have sequence-specific origins, a yeast called Euroia lipolytica. And when Gerry was at a meeting at Cold Spring Harbor, I remember talking to him and asking him, did he know anything about this yeast? And of course he did. And he told me an interesting story. This yeast is 300 million years away from Saccharomyces. So here's Saccharomyces at the top, and here's Euroia down the bottom, 300 million years separation. And it's about the same distance as humans to roundworms. And I mentioned this to Gerry that we were thinking of working on Euro, and he said, oh, it's a very interesting yeast. It's used for industrial purposes. And the coolest example is that you saw from Jeff about the expression of the carotenoid pathway in yeast. Well, this had been done in Euroia many years earlier, and the purpose of it was to make orange yeast like this. This is, came from Gerry's story. To make orange yeast to feed to farm-fed salmon, because the salmon are white, and of course people don't want to eat white salmon. They need salmon-coloured salmon, and you feed them this yeast and they get this colour, because the farm-fed salmon can't go out into the ocean and eat krill. So I thought, well, this is going to be cool to work on. And so we've started working on this yeast, and it turns out that the initiation of replication in this yeast is completely different from Saccharomyces. Almost everything, except for the proteins involved, the origin of replication is completely different. How ORC and CDC6 function is very, very different. And we're now looking at other yeasts to see how quickly this process of a fundamental process of life has evolved very quickly in these yeasts. Now, you heard from Harold about Long Island scientists. I remember we have symposia at Cold Spring Harbor, and the annual symposium, and it's a tradition that we take scientists out to the houses of our significant donors and people of significant means. And one time we went out to dinner, and I was driving home, driving some scientists back, and Gerry was sitting next to me in the front of my car, and in the back seat was Chuck Scherr, who also grew up on the North Shore of Long Island. And we were chatting, and Gerry turned around to Chuck and said, I remember driving you on Long Island one time, and you were dating my sister, and you were sitting in the back seat. And and Rosa, oh, it was your sister. All I had a vision of Chuck Scherr doing something in the back seat of the car. So there are other scientists who came from Long Island. And so with that, I want to thank Gerry. Thank him for all the things he's done for Cold Spring Harbor. Thank him for everything he did for the yeast field, because it has an enormous impact on science if you think about not just the things that came out of Gerry's lab, but all of the people that have gone through that course and have gone on to do major, major things in fundamental biology. And also been involved in starting an institute like this, and then really promoting teaching as a real art. And so thanks very much.
Thank you, Bruce. So now it's my pleasure to welcome up former Whitehead member Terry Orr-Weaver, whose lab was on the 5th floor with Gerry's for many years.
I'd like to start by thanking Ruth very much for the invitation to come back, and for Gerry for the occasion. It's really wonderful to be back and see old friends. And let me just take care of this for a moment. Okay. Okay, and I feel like really at the outset, after Jeff's talk this morning, I need to reassure you all, the theme of my talk is not religion. The theme of my talk is wisdom I learned from Gerry Fink. So Gerry recruited me to the Whitehead as an assistant professor, and was a key supporter of mine throughout all the decades I was here, and I learned a lot from him, and I want to share some of that with you. It allows me to tell you some of my favorite Gerry stories. So when Gerry was recruiting me, and we were talking about my coming up to interview, I was initially very reluctant, and that was in part because the Whitehead had developed a reputation in the Drosophila field as being hostile to Drosophila and to genetics, and this turns out to have been an unfair conclusion. But Gerry reassured me by telling me that, whoops, oh, I'm going the wrong way, that the squeaky wheel gets the grease. And so what he argued to me was that he had been squeaking for a long time and had really convinced the senior faculty at the Whitehead that Drosophila was a great model organism and we should have a Drosophila group. And so I never myself learned to be squeaky, but I'm very grateful that Gerry was squeaky and that I came to interview and then took the job that resulted from that. So after I'd been at the Whitehead only a few weeks, Gerry sat me down and explained to me the meaning of the word meshuggah as a noun. And he illustrated it for me by an example. And I think it's a statement about the caliber of my colleagues at MIT and the Whitehead that never in my 30-odd years here did I ever consider using this word. So those of you who don't know what this word means, ask Gerry during the mixer. Probably best not to ask him for an example, although with the current political landscape, he could probably give you a lot of examples. Okay, so then I think at my first Whitehead retreat, which then was on the Cape at a very swanky place called the Chatham Bars Inn, Gerry and I were seated for dinner at a table that included a lot of the luminaries from the biology department, intimidating people, as well as people on the scientific advisory board. And what Gerry said to me is, you know, in setting up your lab, you really need to do the blood in the water test. And what he meant by that, as he explained it, was the test, it's an experiment, is if there's blood in the water, do you, as a scientist, swim towards it, or do you decide you're going to go to another part of the sea and be there? And before he could even finish, several of these luminaries said, it's obvious, of course you go to where the blood is, because that's where the action is and where it's going to be the most exciting. But Gerry just gave me a smile, and I knew we were on the same page, that I didn't want to go where the blood in the water was. I wanted to go somewhere else in the sea and really look at problems that weren't really heavily investigated yet, like what controls what the sister chromatids do during meiosis I, and what happens during development to cell division in the cell cycle. Are there cues that really alter the cell cycle? And then, as Ruth mentioned this morning, he later articulated this philosophy very eloquently in this autobiographical article that he published in Genetics, which I really encourage you to look at. This was in honor of his being awarded the Morgan Medal by the GSA for lifetime achievement in the field of genetics. And so, as you heard this morning, he put this in the context of stages of a chess game, the opening game, the middle game, and the end game. And this is the quote. He said, "The opening game, a project where little is known, is tailored to my disposition. I'd rather be a tiller of new soil than a harvester from mature fields." I felt very supported by Gerry of wanting to take the same kind of approach. And one thing that he went on to say in that article is that it impacted how he mentored. And you heard about that also repeatedly this morning. What he said was, my taste for the opening game also influenced my mentoring of the many adventurous and talented students who were similarly attracted to this style of research. The project my students developed was theirs. Once they left my lab, they were free to take those research projects with them and to advance them in their own independent labs. In this way, my students' research flourished, gaining them distinction for their own successes. And so I very much wanted to do this too. And as you might imagine, this wasn't considered a winning strategy for an assistant professor, but I was very glad to have Gerry's support to be able to have my postdocs take their projects with them. Okay, this is another bit of wisdom from Gerry, and this came about when we were teaching the graduate lecture course on genetics to the first-year graduate students at MIT. And as we were walking together to the lecture hall one day, and I was going to give the lecture that day, Gerry said to me, "How are you feeling?" And I said, "Well, you know, not so good. I'm pretty nervous." And Gerry said, "Well, I've been teaching genetics for 20 years, and I still cannot sleep on the night before a lecture." So that was kind of sobering, but as many things that Gerry taught me, it was true. Never gets easy. Okay, and this was another thing Gerry taught me. Indirect costs are a real thing. Now, the way this came about was when I got my first NSF grant, NSF had capped the indirect cost rate at 45%. And so if your institution had a higher indirect cost rate, let's say double that, for example, then you either had to, your institution either had to agree to just forfeit the remaining percentage of indirect costs, or it had to come out of your direct costs. So I heard that I had my first grant from NSF, and I heard that the indirect costs I was going to receive were 45%. So I went to Gerry, who was then a director, and I said, "What should I do?" And he said, "Well, you should call up Dalil Nasser and tell her that Gerry Fink, the director of the Whitehead Institute, said indirect costs are a real thing." And Dalil Nasser was this force at NSF. I hope some of you had the chance to know her, who launched so many projects by her support for genetic approaches in model organisms. She really had an impact. So I did what Gerry suggested. I called her up and I said, "Thank you, Dalil, for this grant. However, there's a problem about this 45% indirect cost rates, and Gerry Fink said I should tell you that indirect costs are a real thing." And I don't think I ever told you this, Gerry. Dalil just burst out laughing. And she said, "I can't tell you how funny this is, because all those years on the other side of the coin, when Gerry was running a lab at the Whitehead before he was director, every time I awarded a grant to him, he used to call me up and complain and tell me how outrageous the Whitehead was being with these exaggerated, exuberant indirect costs." Okay, so the other reason, the main reason I wanted to tell you this story is, as you know, this is very timely. And Gerry, as an educator, now that you have more time, I'm thinking maybe you can explain this to Musk and the Musketeers. And I'd really like to be a fly on the wall when you do that. All right. Here's another truism. This isn't a quote, but it's a truism from Gerry, and that is, if you lose a bet, pay up. So the story behind this is after the Broad Institute, after the construction of the Broad Institute was finished, but before any scientists were occupying the building, all of the Whitehead faculty were invited to come over to the Broad Institute for a tour and a dinner. So Gerry and I shared an office suite, and we were chatting about this, and Gerry said, "Isn't it wonderful that the Broad Institute is having us over to thank us for how we helped the Broad get started?" And I was just feeling very cynical, and I said, "You think that's really all that it's about? You don't think that there's a bit of gloating in this?" And Gerry said, no, it's just they want to thank us. So we sort of went back and forth about this, and Gerry said, "I'll bet you $20 that the sole intent here is to show gratitude to us for how we helped the Broad get started." So, okay. We went over, we had our tour. It's a beautiful building. There's a fabulous auditorium on the first floor, kind of like this. All of the faculty offices are surrounded by 4 clear glass walls. That's another story. But afterwards, we go to have dinner in this conference room, and it ends up that Gerry's sitting directly across the table from me for dinner. But mind you, this is not a narrow table. This is not a table where you can reach across with your arm. It's a very wide table. So we're having dinner, and about halfway through dinner, Gerry makes eye contact with me and reaches into his pocket, pulls out his wallet to pay me my $20. So if you lose a bet, pay up. Okay, and one last story. Not long after I'd been elected to the National Academy, I was invited to this summer Sunday afternoon party at an estate on the Cape. I think it was in Woods Hall, an estate that the Academy owned. So I took my invitation and I went to Gerry's office and I said, I've been invited to this National Academy party. Should I go? And he said, of course you should go. You'll meet many famous scientists who you thought were dead. And this turned out to be true also. So the last thing I'd like to do is just point out that through all of his successes and achievements, Gerry never lost sight of the human aspect of doing science and doing research, and cared about all of us deeply, and including what was happening in our personal lives. And this is another great autobiography that has also been mentioned earlier. But at the end, he talked about the sources of support. And the first group he talked about, which I don't have a quote for here, were the technicians in his lab. He specifically mentioned Cora Stiles and Paula Grasaffi, who drove a lot of the experiments, but also were very important in creating lab culture. And then he talked about families, and he said, our spouses, often not scientists themselves, provide the connections between our scientific devotion and everyday life. They are our companions in our success and balm in our failures. And so one thing that hasn't been mentioned today as a gift that Gerry gave to us was sharing his family with us and letting us get to know Rosalie, this truly extraordinary person. So I just want to stop by thanking Gerry for everything he taught me and for being behind me, even in really tangible and concrete ways, helping me through a life crisis. So I'm very, very grateful to him. Thank you.
Thank you, Terry. That was wonderful. Our final speaker of the session is Michael Rosbash.
Thank you very much. It's a pleasure to be here. Thank you, Ruth, for the invitation. I must say, I perhaps know Gerry the least of anyone here. I was never trained by him. I have a few tangential connections, but you have to step up when your time has come. So here I am. So this somewhat illustrates the conundrum I'm in, and I should say I chose this quote in part because my family accuses me of being a Philistine,
And I try to convince them, no, I'm really very sophisticated culturally, and that's a manifestation of this. I should also say that I accepted, I travel relatively little these days, and I accepted the invitation because, of course, it's 10 miles. And also, it is incredibly rare that I get to speak at an event where 3 of the other speakers are actually older than I am. And so, like the remark about the National Academy of Sciences, it is very good for one's self-esteem. I'm an admirer of Gerry's, both up close in ways that I will try and elaborate on a bit, and also from far away. He's really, you have all enumerated the contributions he's made to science. He's a remarkable mentor. And I can't add anything to what's been said. And finally, my last reason for being here is a bit like it's a Yogi Berra quote. If you don't go to your friends' funerals, they won't come to yours. And I've never actually used that joke in a funeral, and I'm not looking forward to having to test the audience under those circumstances. Actually, the last time I used that joke was at David Baltimore's 80th birthday party. So it's a pleasure to do it again. So, like I said, I'm an admirer of Gerry's, and I'm a local neighbor, having worked at Brandeis for more than 50 years. I started to work on yeast in the mid-late 70s, and Gerry's work at Cornell, transformation, et cetera, was really foundational, without which we couldn't have moved forward. So let me say a little bit about this. I was interested in the regulation of gene expression, and Lena Hereford came to my lab in the mid-70s or so, and Lena had been a postdoc of Lee Hartwell's in Seattle. And the yeast community in those days was very small, and so Lena was really embedded in the science of yeast and in the culture of yeast. And it was in many ways through Lena that I became aware of Gerry's contributions to all this. And we produced 2 papers, Lena and I, in the late 70s, taking my pre-recombinant DNA skills of assaying gene expression and applying them to Saccharomyces cerevisiae to take a look at RNA distributions and see what we could find. And of course, recombinant DNA came along, and I realized that we had to embrace that technology. And I was interested in the regulation of a coordinate set of genes. I chose ribosomal proteins because they're stoichiometric in the ribosome. So how does this come about? And I won't belabor that story other than to say that I started to isolate mutants which affected this set of genes. And that led me on the path of studying splicing, because these mutants affected splicing, and the ribosomal protein genes in cerevisiae are a very rich source of introns. And that led me, in a way, to Gerry, who then communicated this paper to PNAS, which was the practice in those days, where we could really study an intron and the function of splicing mutants by simply looking at the expression of beta-galactosidase in cerevisiae under splicing control. And so, but something was missing in my scientific portfolio, in my approach. And I heard a talk that Gerry gave in about 1980, and he used this line in his talk. And I thought to myself, well, and this is unusual, if you know Gerry, because he's rather proper. So locker room kinds of conversations is not really his métier. And what he was referring to was the fact that he had tetrad dissection microscope and micromanipulator in his bedroom, or so he claimed. And so often before going to sleep, he would dissect tetrads. And although I had been working on yeast for several years, I actually had never dissected a tetrad and never used that very straightforward and profound approach to study the problem. And so I went and took the yeast course, despite having worked in yeast for several years. And you've seen this picture before, and Gerry and Fred Sherman prominently figured in the middle. And actually, you've seen this one too. That's the second picture. And that's an action photo of Gerry. And those of you with a tiny bit of imagination will appreciate what Gerry is really doing is shooting a jump shot, or pretending to shoot a jump shot in that picture. And Harold and others have already referred to his basketball prowess. And Gerry and I share an interest in sports. And I should say that Harold's story about the audience for their return to high school reminded me when I played football at Caltech, and we had a real team in those days and so forth. I digress, but we played our home games in the Rose Bowl. And that stadium of 100,000 people had about 30 people in the audience. Two girlfriends, of course, this was Caltech. Not many of us had girlfriends in those days. Couple of parents, et cetera, and a significant number of trainers who were taking care of the injuries that were about to occur. So in any case, Gerry and I had a conversation about basketball and sports, and I contend that I am the only person in this room who knows, other than Gerry, who knows who this person is. So Gerry, as a guard at Amherst, played against Jack the Shot Foley, who played for Holy Cross in the same era when Gerry was around. And he had, as they say, a cup of coffee with the Celtics. He was drafted at the very end, and he was a terrific player. And Gerry and I had a long conversation about Gerry guarding Jack the Shot Foley. And so anyway, this is a phase of Gerry's career which is also notable. So you've all talked about this at some length, the importance of community, the people around you, and so forth. Well, when I went to Brandeis, in addition to Lina in yeast, there was my good friend, pal, Jeff Hall. And Jeff is, to some extent, a segue here in two directions. One is he was also an Amherst undergraduate, graduating in, let me think, 66, I would say, a year after I finished at Caltech. And Jeff was a PhD student at the University of Washington, as was Lina. And Jeff always claimed, I've never actually verified this, but Jeff claimed that the University of Washington had the first genuine genetics department in the United States, started by Herschel Roman. And Jeff and Lina were students in that, and the two of them brought me genetics at Brandeis, Lina in yeast and Jeff in flies. And it's really through Jeff that I got involved in circadian rhythms and fly genetics. And I should just say that working with Jeff for almost 30 years together, our two labs fused, really is what gave birth to this conserved, small regulatory system which governs circadian timing, 5 proteins which are conserved between humans and flies, 600 or 650 million years of evolution, and that small circuit is what does circadian timing, and I'm not going to say anything more about that, although I'll use it as a segue, especially for younger people in the audience, although I think the average age here is significant. this is a slide I show often, and the question is, if this problem is so well explored, why should anybody even consider working on it? What's interesting today? And I want to simply raise this for you and tell you maybe a drop about the second part of this slide. So first of all, circadian is circadia, about a day. So the endogenous period in animals is from 22 and a half hours to 26 or 27 hours, depending on the species. And the question is, what's rate determining? What determines the rate? And the answer is, you have to do biochemistry to really do this. You can't, from the components or from genetics or in vivo experiments, actually determine what's rate limiting. And that is being done at the moment. The second companion question is, why is circadian timekeeping temperature independent? And I'll simply tell you that if you measure the circadian period of a fruit fly, for example, at 18 degrees and 30 degrees, there's no change in the endogenous period. So by that, I mean the Q10 is 1.02 or something. It's almost exactly 1.0. And that's essentially unprecedented in physiology and biochemistry. Everything goes faster with temperature until, of course, you denature the system. And the cell cycle goes faster, really, everything you measure. And I should say that this is not just a property of animals like fruit flies, but it's also true for mammals. And so, of course, in a mammal, you can't do the same kind of experiment because our temperature only varies by a degree Celsius or so. But if you take a tissue out of a mammal and put it in culture or a cell culture system and measure circadian period and do it at 10 degree Celsius difference, there's no change in period. So it is almost certainly, by that description, an intrinsic feature of the timekeeping system, which is conserved, as I mentioned, between flies and mammals. And so I think a combination of structure and biochemistry, which we are working on, and I will not expose you to, will solve this problem. And I think it's an interesting challenge for the future. So returning to Gerry, I think the big theme here today, and it's been touched on in various ways, and is really family, the Whitehead, MIT, and I think just to end on, someone mentioned the word religion earlier, and I think to some extent, some features of these substantial philosophical themes are religion, or for many of us, religion. This job we have, this community, this environment we've been in really gives us purpose in a way, and we have, speaking now for the senior members here, we have received so much from this, from Gerry and from the Whitehead and from MIT and from our environment. It's really been a wonderful journey, and I think it's been a great career in life. Thank you very much.
I'd like to thank all the speakers today for their wonderful reflections on how their lives have intersected with Gerry's life and the impact he's had on them. We're going to take a short break now and be back at right around 3 o'clock.
So I was really surprised.
If you block beta oxidation, if you totally block it, those are also really good. So we know you need to have beta oxidation. We don't know beta oxidation is what. You know, is it fatty acids, is it oxide, or is it adenine, but or something else, right? Because they all kind of go through that same pathway. It's highly consensus, but
And I trusted
Yeah, he told me that. Well, it's great for building. I mean, like at MIT, clearly you have this whole climate initiative, and so I get called on as now one plant person. Now there's one other person. I get called on as plant related, which has its downside. It's like I don't know, I'm not an expert or anything on all these plants, but I mean, I ran a faculty search here a couple years ago, and one of the things we put in the ad was plant biology. There's one, interested in having another plant here.
I think you got to go all in. You should collaborate with JCAS, for instance. It's a fun university, and the research is really very good. I've had amazing opportunity. I've hired almost 55 tenure-track faculty in 5 years. It's really impressive. And they're very good.
Thing you're doing, so you came up with this hiring people straight out of grad school. Yeah, it's really interesting.
Transformative, it totally changed the pipeline. I doubled the percentage of women in math and physics. The total percentage of women in math.
Thank you.
They're up 20% now. I think you're welcome.
Yeah.
And I 45% like women, 40% racial minority, and they're really good, and people are really into it, and all the hires are doing really well. So, yeah, it's been totally transformative. I could never have done it here. I wouldn't have got permission at all. But they took a chance there. And it was so successful that I kept getting more and more slots to do it. And then the other colleges started copying, and now the other universities, Susan Hoffman says refers to the postdoc pipeline, postdoc bubble, which is like I mean, it was transformative. And the only way we did it, we framed it, that we normalized the time of entry into industry and time of entry into the academy. That was what it was advertised. That was so there's nothing there about diversity or anything. It was really what you could do after your PhD. I really worked amazingly well. Yeah, I was lucky. I know, and then we rebuilt PhD training. I totally rebuilt PhD training. So we sort of do it more like medical school. There's cohorts across the whole College of Science. And they make this really strong community. Then they go into their departments, but they make this unbelievably strong community, and they get all these sort of flexible skills. So the kind of lonely road part of the PhD we got rid of. And it's unbelievably popular. The students absolutely love it. And I tell them this is like medical school training we're going to go through as a cohort. So I couldn't have done that here. And I couldn't have given Jennifer the opportunity here either. You would never have gotten that.
How are you doing?
I'm good.
Good to see you. You look great.
Like it. I could innovate in it, and it's been unbelievable. I can tell you sometimes.
So it worked.
Unbelievable. But I've kind of been here 5 years, and I figured I really didn't need to go there. So moving. I started higher education into the most higher education there is. Right now. So I'm moving to UMass Boston and studying at the College of Science and Mathematics.
You know, institutions like that really make change. That is special university surrounded by the privates. And I think I'm hoping that these spokes of a wheel, you know, how supportive is the state? I was
well, see,
just keep making trouble and change and things like that.
How are you? Doing good? You really have big now, huh? How many people?
It's not huge. It's like bigger than it used to be. I mean, I don't want it to be double because I know what's going on.
Yeah, you're involved in a lot of things. Well, that's good. That's good. Now your family? How many kids do you have?
Just 2.
How old are they?
They're 13 and 9.
Oh my God.
Yeah.
Wow. Time is flying. And I was on board for just about 15 years, just right around when you started. And now I'm not anymore. But I know a lot of people don't know that. They even send a note around, hey, Margaret's gone. I just in last year, end of June of 2024, which was kind of bittersweet for me. You know, we passed some bylaws which kind of said, well, you have to be on for so many years. But also, I guess it's time to move on and said now I can sit here and just listen and play around and no responsibilities.
Well, I'm glad to hear that.
Oh, this is really good. I love Gerry.
So you met him and tell me about that.
Very special. Anyway, good seeing you.
That they stay as a cohort, and they get mentoring. They're really a tight group, and so they're so proud. It's one of the great science communities, and they sort of travel together. But it's incredibly popular, so I'm real supportive. So
I remember graduate school for me was like wandering in the desert. Exactly. So, oh, so you were quite intentional on this lonely road to community life. Oh, I like it. That's so cool. All right, so you have 5 years, you'll fix UMass Boston. And then what, a lot of things remain to be fixed, by the way.
UMass Boston will take a while, but I think public higher education is really interesting. I like the UMass system a lot. I'm really proud to be part of it. And I think UMass Boston is at this lovely level of having achieved our status, but just. So what I think
Thank you for doing it. That's so tough. That's so cool.
Yeah, no, it's really fun.
Wow.
You doing okay?
Having a good time? Yeah. What's going on? Yes, absolutely.
Alice found you very nice to work with, so I know why I just talked to you.
Oh, no, there was one person who were trying to fire. I decided to go to the press and some other effect. It makes no difference. It was Newport, Washington, trying to fight.
Oh, no.
So you just sort of like,
Right? Professional, positive.
you just say nothing.
Keep going.
Exactly. Exactly. Absolutely.
All right, well, nice to
Very good.
I'm just moderating. Okay. If everyone could get a seat, we'll get started with the afternoon session. Okay. My name is Peter Radine. It's my pleasure to moderate this afternoon session for this really terrific event. Just a quick remark. When I was an undergraduate, I started a passion for genetics and using genetics to study model organisms. And I remember learning about this famous yeast course and Gerry's role in this yeast course while I was an undergraduate. And little did I know I would be a colleague of Gerry's for 20 years up on the 5th floor of the Whitehead, and then teaching genetics at MIT together with Gerry for over a decade. It's really been a wonderful experience, been a fantastic colleague and friend. With that, I will invite the first speaker up to the podium for the session, Susan Whitehead. Susan is a founding member and life trustee of the Whitehead Institute, and a life member of the board of trustees at MIT. Susan, welcome.
Hi, everybody. So many familiar faces. This is such a sentimental occasion for me, so I can't imagine what it is for you, Gerry, and you, Rosalie. I have to kind of laugh because this is a little bit of a love fest today, and that is not a descriptor that I'm accustomed to using when speaking about you. But, but it is so clear. It is so clear that you are adored and respected in this room in ways that people feel very, very deeply connected to. So it's been a pleasure to see. So how would I describe you as director of Whitehead Institute and how you led here and who you are? There is nothing inconsistent in anything that I am going to say here. They're all the same threads throughout everybody's remarks. You're wise. You have the highest integrity. You have very simple, straightforward human decency, and that matters to you. You're a steady force. You're very measured. You're very calm. You always knew the pulse. When you were director here, there was nothing that was surprising to you. You had already heard about it wandering the halls. You knew what everybody was thinking, and that really, really made a difference here. You knew people's capacities. You knew their limitations. You really knew people. You have the highest expectations. You're very discerning. You're very fair. And you had the kind of vision when you led here that does not call attention to itself at all, but it's rather what I would call a kind of shepherding of your flock so that they could all do their best. Your presence was not an obvious, overwhelming presence, but you were unfailingly encouraging. So what are some of the highlights of your time as director here? What are you all looking at? I remember these wonderful pictures. So I imagine that others will continue to speak about your time as a leader in science and with the faculty and with the fellows, but I'd like to talk about some of the institutional things that you did here. And as I was putting this together, I realized you did so much, but again, to go back to your character of leading, it wasn't obvious all the time that you were doing so much, but you were. So what were some of those things? First of all, I just want to say out loud that you had us look decidedly and deliberately outwards in a way that we had not done as an institution before. And, you know, we had done some things, and David certainly had done some things and been in the public eye, but in a very considered way, you developed programs that had Whitehead in service to the public, educating the public, and certainly exploring public policy issues, all the time. So what were those programs? We have a great high school teachers program where teachers come once a month. They do this for a year long, and they partner with a trainee at Whitehead, and they learn about the forefront of biology and how they're going to teach their high school biology students. It's been very popular. It's about 60 to 80 high school teachers a year. That was a big deal to establish. You also established a high school teaching program where students could come during vacation time and could study here. You also, I did want to say something about your written materials. I mean, everybody here knows Gerry is an incredibly good writer. He is so precise. He loves language. He loves a great turn of phrase. He loves the craft. And all of the materials that came out of Whitehead under his tenure were really quite remarkable. Okay, talking about the outward-looking things. You did these policy symposia when nobody was doing that. And you did two very important big symposia that were in Kresge having to do with policy, ethics, technology, how we were supposed to think of health, science, and policy in the 21st century. Those were very well attended, really well liked. But you also did a whole bunch in collaboration with David Page, I'm trying to see up there. There he is. And Eve Nichols. We did all kinds of workshops at Whitehead Institute that frankly were things that would remain with me to this day. It was 25 years ago. And I remember talking about how our parental rights law was based in property law in the United States, and you could trace back to property law principles. Remember talking about paternity and the untruth of paternity in question, and what public policy was served by making that known and elucidated. Was there any value in making that known? We had another wonderful session when we talked about the beginning of life and how it interfaced with stem cell research. What was the definition of the beginning of life and how you were supposed to think about that coupled with public policy decisions that were being made right in the moment. These were all just marvelous, educated the public, completely outward-looking. We also did symposia for several years, educating the science journalists at the major science publications and major newspapers throughout the country. They were highly valued. Okay, I would like to switch gears a little bit and talk about the board, because you had to deal with the board of Whitehead Institute. And so many of you here know a lot of people on the board, and you know about the board. It was at times kind of an unruly bunch. It wasn't the most straightforward board to organize, and Gerry was really effective with the board. I recall so well your delight when you recruited Paul Sangas to become our chair. Paul was utterly stunned that you had invited him, and very, very happy. He would oftentimes say that he thought he owed his life because of his cancer. He'd owed his enduring life to the kind of work that was done at Whitehead Institute. So he was very clear about that, and it made him completely curious and a very effective and enthusiastic booster of all things. He was also quite good with the board, and he led with a certain dignity and gravitas, which I think was very effective during those years. So, let's talk about the Genome Center. I know that we'll be talking about the Genome Center in some detail in a little bit. So I'm not going to talk about all the details of the Genome Center, but more your role in it, Gerry. It was a really big deal to have the Genome Center here. Again, take yourself back 35 years. This was a completely new kind of science and scale. and scope and equipment and cost and speed. There had been nothing like it, certainly in the life sciences, ever before. And it required a lot of courage on the Institute's part. I recall very clearly when Eric needed to get some sequencers, and he needed them now, and it was going to cost us, it was sequencers and other equipment, it was about a $35 million outlay in order to get those sequencers. And you might say 35 million, that's okay. That was half of our annual operating budget. And there was there was no guarantee from the government that we would be repaid that money. It seemed pretty likely that we would be, but there was no guarantee. And Gerry, along with, in partnership with John Pratt, okayed that and moved that through the board. I did mention the unruly board, and so that was no simple task. There was a lot of heated conversations about that decision, but Gerry was absolutely unambiguous about its importance and its significance for the future of science, and that we had to do it. Gerry also had the vision and foresight to expand our building. And again, he had to go to the board about this. He wanted to expand it by 70,000 feet, 70,000 square feet. That was also a big deal. And you have to, again, put this in some context. We were only 10 years old, and so there was resistance from the board. We're 10 years old, and you want to expand us by 50% already? We felt very comfortable with the assets we had and the liquid cash that we had. We could very freely seed projects that we wanted to seed, and that was a great strength that was very valued by everybody who worked here. And some of that was going to get whittled away. And so Gerry had to go to the board. He was certain that we needed more space, and we recruited just spectacular junior faculty whose labs had grown unusually quickly. They were so successful, and they just needed more space here. So he was quite convinced, and he went to the board. Many of you in this room might remember Abe Siegel from Sloan, who was the dean of Sloan, the wonderful dean of Sloan. And Abe was a great asset in your going before the board, Gerry, because I remember very clearly that Abe said, you know, an institution that doesn't grow and evolve will not thrive. And Abe was our trusted elder, and he really occupied that role on the board. And so between your tenacity and your conviction and Abe's very persistent persuasion, we did that. I just want to point out, we raised $13 million. We had never done any fundraising, any organized or formal fundraising before. We had no fundraising arm. But Gerry believed we could do it, and lo and behold, he was absolutely right. Part of it was fun, and part of it was community building, and there's just one story I'd like to tell about that particular era. Paul Tsongas was our chair, and we had to go out to Michigan. Gerry remembers this story very well. We had to go to Troy, Michigan to visit with the Kresge Foundation. It was a big deal. I think we wanted, out of the $13 million, I think we were asking them for if they would consider $3 million, something of that scale, in supporting us. And so we go out, and it's quite in the middle of a quite quiet place. So we walked in, it's me, Gerry, and Paul Tsongas. And it was like going with a huge celebrity. Everybody wanted Paul's autograph. They wanted to speak with him, and we just were not anticipating this. And it ended up being an awful lot of fun, and I might add, a successful meeting. So it worked out in the end. So to close, I want to say that personally, I so enjoyed working up close with you, Gerry. It's hard for me to separate out with Whitehead Institute the personal and the institution in some ways, but you know a number of my family members, and I'd say they're all loud, they're aggressive, they're very sure-footed, they take up a lot of space. And Gerry, it was so interesting for me to be able to see you, who are the complete opposite of all those things, work so effectively over time and build a relationship with all of them. And you did it really, really, really well. So thank you. Your period when you were director was a very happy period at the Institute. And I believe it was all because of the quality of your person and the integrity you brought to everything and the fairness, and that people were really, we heard it all throughout today, people felt very safe with you, they felt very revealed to you, and your care for this institution was clear always, all the time. So thank you.
Thank you, Susan. That was wonderful. So our next speaker is Eric Lander, founding director and core institute member at the Broad Institute. Of course, before founding the Broad, Eric was a faculty member at the Whitehead, and before that, a fellow at the Whitehead. And today we're going to hear about Gerry and the genome. Eric.
So thank you, Peter. So Susan said a little bit about this, but I'm going to say a lot more about this. So yes, I joined the Whitehead as a fellow in 1986. David Baltimore recruited me and became a faculty member in 1989. And the very next year, Gerry became director. He stayed director through 2001. That happens to be the exact period of the core of the Human Genome Project, which started in 1990, the year you became director, and reached its really most important culmination in a draft sequence of the human genome in 2001. Coincidence? No, I don't think it's a coincidence at all. Gerry played an absolutely central role in the success of the Human Genome Project, and it's a story that's not told enough. I've told it occasionally, but not lately, and it's really important that everybody understands that without Gerry, there was a very reasonable chance we would not have gotten a genome project done. Let me try to defend those statements, because I deeply believe them. The Whitehead Institute ended up being the flagship of the Human Genome Project. It was the place that contributed the most to the Human Genome Project. It was, at the end, a tremendous international force. There was nothing about that was preordained. There were serious medical schools and universities who were all gearing up to be the driving forces. But the Whitehead had a superpower. Actually two superpowers. The first superpower was it's an independent research institute, a smaller independent research institute that can actually, if it's got wise leadership, understand the science and make good decisions about the science. And of course, the second is that it had amazing leaders. David Baltimore, of course, as our founding director here, and then Gerry, who stepped in at this crucial moment and brought what only, I think, a place like this can do, which is apply institutionally scientific taste, the ability to make judgment, and the ability to take risks. It's very hard for big institutions to do that. go imagine being the president of Yale, which you know well, or Amherst, or Princeton, or Harvard, or MIT. You don't really get to use your taste because it's such a vast canvas. But the ability to actually bring deep scientific taste and then place bets is a very special thing. The existence of the Whitehead created the permissive conditions, Gerry might say, as a yeast geneticist, but it took somebody like Gerry to actually do it. Why was Gerry the perfect person at this time? Well, Gerry is a geneticist. He understood that the human being was a benighted organism compared to yeast. And it was necessary to do something about that. And that someday humans might aspire to the scientific standing and stature of yeast. That we might know enough about the human to be able to do like real experiments as opposed to the potchkeying around that would happen in a medical school or something like that. And he had that vision from the beginning. The first time I talked to Gerry about it, he knew that's what this was about, was turn the human into yeast. This was not easy to do. I will tell briefly the story in 3 acts. Now, we had many scientific discussions about this. I'm a geneticist, he's a geneticist. We had lots of genetic discussions. But since this session is sort of leaning toward the institutional, I want to talk about the 3 big institutional things Gerry did. Number 1, 1990. We needed some place to do the Human Genome Project. We'd gotten a little bit of real estate on the first floor of the Cancer Center. I mean, 2 rooms, not very big rooms, but 2 rooms to get going on a little proto-Human Genome Project. But then in 1990, Human Genome Institute is founded, the Human Genome Project starts, and we want to apply to be one of the first genome centers at the Whitehead. It was not going to fit in these 2 little rooms. So of course, because we were doing this over some rooms in MIT, I went to MIT first and I saw the provost. Those of you who are old enough will remember that at the time, the provost was John Deutsch. Some of you remember. So I visit John Deutsch. And I explain Human Genome Project, space, we need this kind of thing, really important, et cetera. And John looks at me like only John could do, and he says, "You want space? I don't have space. Maybe I can get you the revolving restaurant on top of the Hyatt on Memorial Drive." I've never forgotten this. As I was briefly contemplating whether we could do the Human Genome Project while rotating or something like that. But I think what he meant was, I'm not going to be able to do anything for you. And I was somewhat depressed because I thought we stood a chance of actually building a really great genome center at a place like the Whitehead. So I came back to Gerry a little bit, I had been on the faculty less than a year when I had this encounter with John. I've since learned to maybe, interpret John better, but I came back to Gerry a bit shaken by this and said, "Gerry, what can we do?" And of course, this genome center is sitting at MIT at that point in these 2 rooms in the Cancer Center, and Gerry does not miss a beat. And he says, "I'll call John Pratt. We'll go out tomorrow and start looking for real estate." He didn't ask a word about the financing of it. He didn't ask how big the grant was going to be. He said, "I'm going to go out and I'm going to start looking for real estate." And they did. And 2 weeks later, there was a total real estate survey of Kendall Square. And there was one place identified at 1 Kendall Square, just a block over there, the 300 building, that some tech company had just vacated the 4th and 5th floors and was available. And Gerry decided we would enter into an option on that space, putting some money forward to buy an option so we could do it if the grant came through. The grant came through, and we had a genome center to start with there. And I put to you, there is no institution I know other than the Whitehead that would have pulled something like that off. And that was the first foundational thing. So then we outgrew this space. Now, admittedly, it was the 4th and the 5th floor, but it's not like that big a floor plate. And we did the physical mapping and things of the genome there, but still was not enough. We needed a serious warehouse to do the Human Genome Project in. So I saw Gerry, and Gerry, again, did not miss a beat. He said, okay, how much space do you need? I said, well, we need a lot, but we don't need it all right now. So we need a lot, and we need to be able to grow into it, et cetera, but we couldn't pay for all of it now, et cetera, et cetera. I'm vaguely, at this point, understanding how you finance buildings. I didn't understand much about that at the beginning. And Gerry calls John Pratt, and the two of them go off and do a real estate survey of everything that's available. And they find, sitting over on Charles Street, just a couple blocks up, 320 Charles Street, which was a beer and popcorn warehouse for Fenway Park. It was Boston Concessions. And it truly was a beer and popcorn warehouse, and it served Fenway Park and some other kinds of sporting venues and things like that. And these beer trucks would drive in and float up with beer and drive off and all that. And it happened to have been owned by the real estate developer Joe O'Donnell, a well-known person in Cambridge and at Harvard. And they talked Joe into it. Now, it helped. Joe had a child with a genetic disease, so he understood the importance of this. But it took a Whitehead Institute finding Joe to give us a third of the beer and popcorn warehouse. So we got going in the first third of the beer and popcorn warehouse while the trucks were still driving in and out, delivering beer from the rest of it there. And then over the course of the next several years, they exited more and more as we were able to occupy it. And we took over the entirety of this former beer and popcorn warehouse. And well, that was where the Human Genome Project got done. It would eventually get done in that space there because, well, Gerry knew that it was genetics, and genetics was important, and someday the human might live up to yeast. Act 3. Susan mentioned it briefly, but I'll tell you anyway because it's a much richer story even than Susan told you. Late 1990s, the Human Genome Project has been building genetic maps, building physical maps, and now it's really ready to get going on sequencing. And then we have the Great War with Celera Genomics. Craig Venter declares war on the Human Genome Project. He had his own little genome center. They hadn't done that all well, but the sequence maker, Applied Biosystems, came to him and said, we have these new capillary sequences. We're going to give you a lot of them, and you can sequence the genome, and we can have a proprietary database of all the human genes that people can sign up for and pay for and things like that. And Craig in the New York Times said, the public sector should simply move on and do the mouse. We'll take care of the human. They did. Go look it up. So the NIH was not quite ready to give up yet. And finally, a lot of us had been urging, we were ready to move forward, we were ready to move forward. They allowed applications for serious scale-up. And the Whitehead, which by this point had shown its mettle in being able to do this kind of work, ended up with by far the highest score on the grants that were submitted. And we got this grant, and the grant included equipment money, $40 million of equipment money to be able to buy a whole fleet of these sequencers so we could get the Genome Project done on time. It was so exciting. The $40 million came in the following form. $8 million a year for each of 5 consecutive years. The problem was, we needed them all now. We didn't need them coming in little slugs over the course of 5 years. So we went back to the NIH and explained this problem. We need the money now. The NIH said, well, we don't have the money to give you all of that now. We can give it to you in $8 million. I said, yes, I understand that, but that's not going to solve the problem. And so we asked, Could you give us a binding legal commitment that you would do that? And the NIH said, well, actually, we're not allowed to. We actually can only, we get appropriations every year, and we can't give you a multi-year commitment like that. We can only give you the pink sheet you got back that said $8 million, et cetera. I said, huh. So I had to go back to Gerry and explain we needed to buy $40 million of sequencers with nothing more than $5 million in current funds or so. And could you front the money, Gerry? And as Susan has already noted, this was a non-trivial fraction of the Whitehead budget. It would really, honestly, require borrowing $40 million and paying it back and all that. And it was like, I know today $40 million, it's walking around money, but back then that was real money, $40 million. And basically, Gerry, well, he's a geneticist. He knew this was very important. And he said, okay, we're going to figure out how to do it. And he went to the board, as Susan has said, and said, we need to borrow $40 million. And I went to Francis Collins and asked for a letter, says Gerry, promising that they'd pay it. But the best I could get was an extremely warm letter from Francis Collins assuring us that this would be a, quote, high priority going forward, which the board understood not to be worth anything really other than, barring some kind of thing where the NIH stops paying grants and doing things like that, we'd probably get the money. Thank God we did not have current experiences to reason from. And we got it. Now I'm going to say 2 other large genome centers also were part of this at Washington University, St. Louis, and at Baylor College of Medicine. They could not pull this off. They had huge institutions, and they could not see their way to figuring out how to solve the problem. Gerry, because he's a geneticist and knew it mattered, and because he was a marvelous institutional head who took full advantage of being a scientist running a scientific institution, who felt problems were solvable logically if they were worth solving, solved it. In 2001, the draft sequence of the human genome came out as a paper in Nature in February. Gerry Fink is not listed as a collaborator formally, as a co-author of that paper. But I will put to you that that paper would not be there but for Gerry Fink and what he did to make it possible for the Whitehead Institute to be the one institution in this country that could step up and take on a challenge like that. And that's what's so special about an institution like this, and that's what's so special about Gerry. And Gerry, I've told you before, but it's great to get to tell you today that what you have done as a partner for me, for everybody else around here, has been transformative in our lives, and I think transformative in the whole scientific community. And I hope someday, I think we're getting close, the human may really live up to being almost as good as yeast. Thank you, Gerry.
Thank you, Eric. Okay, our next speaker from Berkeley, Professor of Molecular Cellular Biology at Berkeley, HHMI Investigator, Randy Schekman.
Thank you, Peter, and thank you, Ruth. When Ruth issued the invitation to come, I think it was very late for me, and I replied immediately with delighted at being invited to participate. I spent 3 glorious weeks, well, glorious for the East Coast weather in August may not be the right word, in Cold Spring Harbor, learning from Fred Sherman and Gerry Fink about the wonders of yeast genetics. I want to put this in context, though, because it was a period of time in the history of molecular biology when there were limitless opportunities for the future. You know, today has been such a bright day in an otherwise very bleak time that we face currently and in the years ahead. But we nonetheless have all been blessed by the enormous investment in this country in basic science. I'm a child of the Sputnik generation. We had enormous new investments in science and science education in the public schools. In California, the University of California doubled in size during the administration of Jerry Brown Sr. I was a beneficiary of that, along with other children of middle class, of the middle class. I intended to go to university in preparation for a career in medicine. But during my freshman year, I was introduced to molecular biology with Jim Watson's first edition of the Molecular Biology of the Gene, which really changed my life. There are people today, alive today, that can thank me for not having gone to medical school. I had the opportunity in my first year to work in a research laboratory and to explore the mechanisms of DNA replication. This was, for me, a revelation for which I am so grateful. Well, speaking as one of the senior but still vertical scientists here in this session this afternoon, I want to tell you about the background that I appreciated and that I was the beneficiary of before I had this seminal experience in 1976. So let me give you a period of history and how it informed my choices and the journey that I took to that meeting in Cold Spring Harbor. Studying DNA replication in the late 1960s, I was introduced to the use of the ultracentrifuge and separating, replicating molecules. But I became aware of the work of two great scientists in California, one Arthur Kornberg, one of the great enzymologists of the second half of the 20th century, and another Sinsheimer at Caltech, both of whom collaborated in 1966 and 67 to do something remarkable at a time long before one could clone genes or sequence DNA or do immunoblots. There was nonetheless a rich history of molecular biology. So let me take you back to 1967 with my next slide. Yes. So here are two papers published in succeeding months in the proceedings, work done in Kornberg and Robert Sinsheimer's lab, where they used the enzyme DNA polymerase, the first DNA polymerase that was first discovered and described by Kornberg in 1959, to show that it was capable of copying a circular DNA template to make a double-stranded replicative form, and then to copy that to make an infectious nucleic acid. This was considered a triumph of the time, and it was evidence that Kornberg presented in support of his view that this enzyme, DNA polymerase, must be the enzyme responsible for chromosome replication. Now, Kornberg, in spite of his tremendous gifts, which I learned subsequently as a graduate student in his lab, had one serious blind spot. He was opposed to the use of genetics. I heard him once referred to geneticists as lazy biochemists. This blind spot was very unfortunate for him because in the summer before I started graduate school, in Cold Spring Harbor, the then director of the Cold Spring Harbor Laboratory published a paper, John Cairns, before Watson, before Stillman. John Cairns was the director of the Cold Spring Harbor Laboratory, and he had been resistant to the notion that the polymerase was involved in replication. He proved this by isolating from a random collection of mutagenized colonies one colony that grew perfectly normally of E. coli, but which failed on extraction to express the DNA polymerase activity. Nonetheless, the cell showed that the cells were sensitive to ultraviolet light, demonstrating quite dramatically that this enzyme was in fact rather a repair enzyme. Subsequently, in extracts of this mutant, Kornberg's middle son, Tom, then an undergraduate first at Juilliard and then at Columbia, joined a laboratory, the laboratory of Malcolm Gefter, who subsequently was a faculty member here at MIT, and extracted another enzyme which became the replication enzyme, the real DNA polymerase. It was dubbed the Kornberg Jr. enzyme in an editorial in Nature magazine. Now, faced with this really revolution in the study of DNA replication, I still was enthused to learn enzymology from Kornberg, but we were faced with the attempt to identify all the other components of the replication machinery. Bacterial geneticists, Emerita and Jacob at the Pasteur Institute, and another group at Edinburgh had isolated a series of temperature-sensitive DNA replication mutants, about a half a dozen genes. But this was before cloning, so there was no possibility of identifying the genes themselves. And yet, at the time, in my undergraduate education, I became aware of some pioneering work done at Caltech that showed a path forward to use these mutants in order to explore the enzymology of replication. These were experiments conducted by a young assistant professor by the name of William Wood in collaboration with his senior colleague, Bob Edgar. Edgar was a great bacterial and phage geneticist. Edgar had isolated a series of mutations in the genes of the T4 phage, defective in the production of infectious phage particles. Wood, who had just come from Paul Berg's lab as a PhD student, decided to join forces with Edgar and to devise a then novel approach at biochemical genetics to take advantage of the complementing mutations that Edgar's lab had produced to devise a biochemical complementation assay to purify the relevant gene products responsible for producing infectious particles. And I'll show you just one experiment that still makes my heart beat faster because it is simply an elegant experiment, published actually in Scientific American. What they did was to infect complementary mutants in different genes in separate preparations of phage infecting E. coli, make extracts, each of which individually produce no virus particles, but then on combination and incubation, over the course of nearly an hour, a greater than 3 log fold increase in the production of infectious particles was produced. So this was a model of how to follow a complex biological process using the foundation of genetics and then applying biochemistry to understand a process and a mechanism. I applied such logic as a graduate student when I was in Kornberg's lab and was able to isolate many of the replication proteins. Now, at the time, there was also a great ferment in the birth of the recombinant revolution, which was championed by Paul Berg and one of his graduate students, Janet Mertz at Stanford. So there's a great deal of interest in recombinant DNA. But I was focused on other biological processes, and I took note of the pioneering work of Leland Hartwell, who in the early and mid-1970s had used yeast, Saccharomyces cerevisiae, as a model organism to understand the genes that are involved in transitions through the cell division cycle. And in a really very simple, elegant approach, he took randomly mutagenized colonies, much as Cairns had done, and looked for temperature-sensitive growth mutants that arrest at a unique terminal phenotype. So this seemed to me to be a model also of going forward to study more complex biological processes. I chose, by analogy to what we heard earlier, not following the blood in the water, but to swim away from it, to focus my future efforts on biological membranes, because it seemed likely that the approaches to studying the biosynthesis of macromolecules, such as of DNA, RNA, and proteins, which are covalent structures, there would be something entirely different about the biogenesis of a biological membrane, because this is, unlike covalent structures, a fluid structure in which membrane proteins and lipids are free to diffuse about or spin about an axis perpendicular to the bilayer. So there had to be something else governing how these macromolecules could be assembled. I was aware, because of a deep friendship that Kornberg had with George Palade, the pioneering cell biologist, who had uncovered the intricacies of the secretory pathway, studying mammalian tissues and mammalian cells. Indeed, all of cell biology at this time, in the late 60s and mid-70s, was dominated by studies with animal cells and animal tissues, which were really, at the time, not amenable to genetic analysis. So I turned, after my postdoctoral period of studying membranes, to learn about genetics and yeast, neither of which I'd had any experience with in my career. So then I found myself in the August of 1976 at the Cold Spring Harbor Laboratory It was a remarkable time. Here is a picture, a slightly large version of the picture that you've seen with Gerry and Bick and Tom Pettis, who's here, was included in this picture. So a remarkable group of students taking the class. I was challenged. I think I held the record for the fewest tetrads dissected by any student in the course. But I used as my excuse the fact that during that time at Cold Spring Harbor, there was a hurricane that passed right over, Hurricane Bill. The electricity to the lab was closed down for 3 days. And I remember celebrating with Jim Hicks, Amar Klar, and Jeff Strathern, who were teaching the class along with Gerry and Fred, celebrating in Davenport Hall as the waters of the harbor lapped up against the porch behind the building. So we were stuck behind and had instead an all-night drinking party. The course itself was also quite remarkable by the people who were attracted to come to lecture. I remember meeting Lee Hartwell for the first time, Ira Herskowitz for the first time, and the great David Botstein, who made an enormous impression on me and continued to do so through years of yeast genetics meetings at the Cold Spring Harbor Laboratory. Indeed, my first experience at a Cold Spring Harbor yeast meeting, I was sitting in the middle of the audience. Botstein was sitting in the front row, and some person from Europe came up and gave a boring and bad talk, and most of us just turned off. But David Botstein, unlike the rest of us, stepped up, and in a grand gesture, sweeping by the speaker and the audience, he said, "This is an example of bad science." Now, this contrasts with the usual behavior that I see at scientific meetings now, where everybody is so congratulatory and wonderful talk, blah, 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 but Botstein was honest. So I had an enormous and favorable impression of the yeast community from that brief 3-week experience. Well, just in the weeks after I left Cold Spring Harbor, my wife and I took the road from San Diego to Berkeley, where I began my career. Berkeley in 1976, the birthplace of the free speech movement, but was also a ferment of interest in the birth of molecular biology. And unlike my experience with Kornberg, who was, as I say, a great enzymologist, but with a blind spot to genetics, the department chair of the biochemistry department at the time, Daniel Koshland and Bruce Ames, became my close colleagues, and they were very encouraging of efforts that included genetics and biochemistry. So I had not only the great good fortune of a career in that department, a department where I've remained for now nearly 49 years, but more so because of the experience I had with terrific graduate students and postdocs. In fact, I'm quite confident that my career would not have advanced were it not for the opportunity that I had to train people like Peter Novick. Back then, we all had long hair. He has even less hair than I have now. But Peter has gone on to his own tremendous career, and he is the George Pallotti Chair of Cell Biology at UC San Diego. Peter and I embarked on an effort that led to a genetic dissection of the secretory pathway. I'll conclude with that slide. But first, in the year after we began our efforts, Hinnon, Hicks, and Fink did their remarkable work in developing cloning. This became the revolution that swept over all of us in the field. It was possible to clone genes in yeast quite easily at the time. And finally, over many years after that, we were able successfully to dissect the pathway that Pallotti had been able to visualize by cytologic means, but through the intervention of genetics and then molecular biology, we could trace all of the genes in this pathway. But whatever success I've had, I attribute to people who followed in Peter Novick's footsteps, and I have been enormously blessed by the advantage of having people like Ray Deshay, Linda Hickey, David Baker, Chris Kaiser, Scott Emmer, and many others in the years since in my lab. So although I credit them for the work that has been conducted in my lab, I want to credit Gerry for 3 remarkable weeks in August of 1976. Thank you, Gerry.
Thank you, Randy. So now it's my pleasure to welcome up to the stage the man himself, Gerry Fink.
Phew. I don't know if I can take all this. Thank you all. I want to thank Ruth for organizing and her staff. It's overwhelming. I should mention something that I told my students after they came back after a meeting, and that is, don't believe everything you hear. Okay. I mean, this symposium really honors my students. Okay. Many of whom are here. After 50 years of teaching, I subjected more than 3,000 students to my love of genetics. We did leave out, and I have left out, some important people, okay, that form the bulwark of all the scientific endeavor. If you're not a scientist, you can't appreciate this. If I could have the first, yes. Many of you who are in my lab probably recognize her, but if you don't, the next slide. Could I have the next slide? That's my technician, Paula Grisafi. Okay. And the next slide, Cora Stiles. Okay. For every lab, there is technically, but not just technically, they are the heart of the laboratory, and they deserve as much accolade as anyone else. Now, you've heard a lot of good words about me, and you'd think that's, I got what I deserved, okay? But I was thinking as people were talking, I had visited Copernicus' study in Poland. And in his study, they were very proud to show me his shoes. Okay? His shoes were interesting because they looked like regular shoes, but they had a little hibachi-like grill underneath each shoe. I said, what are those for? They said, oh, before Copernicus got up, his students came in, put charcoals under his shoes to warm them so that when he got out of bed, okay, he stepped into warm shoes, okay? We don't demand that of our students. This will be short, okay? Those are usually deadly words in a talk. People say, I will be short, and then go on for an hour and a half. But I will be short. How did we get such wonderful science? If I can have the next slide. Oh, no. Sorry, I'm out of. This is a typical lab meeting, okay? You can see people are having a really good time. And this is where things are imparted that you can talk about, experiments that work and don't work. And they're very exciting meetings, which are held every week. They're exciting for everyone. Next slide. Occasionally, there are those people who don't find them so exciting. So how did we get here? Next slide. In 1945, Vannevar Bush wrote a paper that influenced American science. Science was called Science, the Endless Frontier. Scientific progress is essential to national security and economic growth. It had enormous influence. The government should fund basic research, and universities should be central. This was a covenant between universities and the federal government. Okay? Out of this, the National Science Foundation was born. NIH budget went way up. Okay? It completely influenced universities, this covenant between universities and the federal government. So we should not call these MIT or Caltech or Yale or Harvard students. So we should call them NIH students, okay? Just as we should say, not the Moderna vaccine, we should say the NIH vaccine, okay? Because they wouldn't be there without all the basic science that made it possible. In fact, if you look at the next slide, here are all the Nobel Prizes before 1960, okay? And the United States looks pretty much like France, Germany, and the rest, okay? This is after Vannevar Bush's effect on the government, okay? This covenant, initiated by Vannevar Bush, has now been broken. The NIH can no longer be depended on to honor the covenant, which produced such remarkable medical progress. People ask me, what are you going to do now that you don't have a lab and are retiring? I will do everything I can to fight to restore this covenant, which I think is so important. And I ask that you do the same. Thank you.
Thank you, Gerry. And thank you to all the speakers in this wonderful afternoon session. Now for the closing remarks, we'll turn to the chair of the board of directors here at the Whitehead, Sarah Williamson.
Well, thank you, Gerry, for those inspiring and remarks and fighting words, and I think that is what we need right now. So I can't wait to work with you on that effort. I want to thank the presenters, the speakers. Many of you have traveled far and wide to get here today to mark this occasion. And thank you for sharing your work, your current work, your cutting-edge work, the history of your work. It does bear repeating that the research that we have talked about all day today embodies the very spirit of that covenant, as you put it, and of this institute, its founders, and particularly of Gerry. Each of you is demonstrating your vision and your commitment to the mission of this institute to forge new frontiers in science and to uncover insights that unlock the potential of tomorrow. So Gerry has set a high bar for all of us. He's been unwavering in his commitment to Whitehead and to the scientific discovery for more than 40 years. Our speakers, as well as our current and future scientists, have the benefit of following in his path that he has forged. Judging from the incredible science that we've heard about today, those in this room are already trying to live up to those expectations. So Gerry, on behalf of the entire board of directors, I extend our thanks to you for your contributions to this institute. Gerry, you have steered the Whitehead Institute through decades of growth, opportunity, and success. Your leadership as institute director has cemented the values and mission that you and your fellow founders have embodied. Your vision and the decades of work you devoted to it have made this institute one of the most prestigious, prolific, and renowned research institutes in the world. And I'm sure Jack Whitehead would have been very proud if he were here today. So thank you, Gerry, for everything you've done, not only for Whitehead, but to advance science and disseminate knowledge throughout the world. And at this time, I'd like to ask Ruth to join me on stage as we present you with a special gift from the Institute. So Gerry, can you come up here with us, please? This is a gift from the Institute that is meant to display some of the pictures that we've seen today, but in a way that you pulls from your book all the work that you've done and tries to be absolutely for. So Gerry, congratulations and thank you. Thank you very much.
Okay, so this was quite a wonderful day, and I think Gerry told us once again what we're supposed to do. On a more operative point, I'm going to invite you to the reception now, and also there will be some opportunity because some of you may still want to say something, and so there should be some opportunity also that you exchange your friendship, but also what Gerry has meant to you. And I really thank everyone who was able to come, and I really thank all the speakers. I think I hope that some of what we heard today will give us the strength to do what Gerry asked us to do. Thank you.